All right, so in this lecture, we're going to be talking about mast cells. This is a personal favorite cell of mine because they're just so important to allergic diseases at large. So let's get right into it. So as far as learning objectives go, we're going to talk a little bit about the biology of mast cells, and then we're going to talk about how that biology informs their structure, their function, and what they do in the human body. And we're also going to talk about specifically their role in some clinical areas such as anaphylaxis, as well as how they're activated. So let's just preface before the actual lecture, talk about the general role of mast cells. So we all know that mast cells are very involved in allergies. That's sort of what they're most known for. But I also just want to quickly mention the fact that they have some roles in, for example, wound healing, fibrosis, blood flow, immune cell recruitment, and protection from bacteria and parasites as well. So again, that question comes up about why we have these things that are seemingly detrimental to us, and it's because a lot of the time they have functions that are positive that just don't necessarily get as much time in the spotlight. So let's get into the biology of it. Mast cell development is a great place to start. So they derive from CD34 positive and CD117 positive mononuclear cells. Now that CD117 is a tyrosine kinase KIT positive marker, and this KIT positive is going to be really important as we move on. So like many other immune cells, their differentiation and proliferation is really influenced by the cytokines that they are surrounded by. And we know from the last presentation that IL-9 is going to be one of those big cytokines. But really the one that we're going to introduce in this lecture is stem cell factor, or SCF. And this is sometimes called the obligate human mast cell growth factor. And what that means is that mast cells don't grow without this. They need this. They're obligated to have SCF in order to grow. Now, stem cell factor is also the ligand for the KIT receptor. So if you remember how these are KIT positive, that means they have the KIT receptor, and the KIT receptor responds to stem cell factor. If we remove stem cell factor in vitro in experiments, we see that mast cells quite rapidly begin to die. So they need this signal to interact with their KIT receptor to survive. So as far as phenotypes, we have phenotypes that are characterized by their serine protease content. So as I talked about last lecture, mast cells can be the T phenotype, meaning they only have tryptase, or they could be the TC phenotype, which means that they have tryptase and chymase. So the T phenotype is most commonly found at the airway epithelium, and the TC phenotype is more found at the connective tissue. So just a little bit of interesting experiments have been done to show that stem cell factor alone is not enough to produce the full TC phenotype. It requires growing the mast cells on a layer of fibroblasts and endothelial cells. So really the takeaway there is that even if we don't know what these fibroblasts and endothelial cells are contributing, we know that there are these other signals that are really crucial for full maturity of a mast cell. It's not just stem cell factor. So mast cells are going to interact with various cytokines. So as I said, IL-9 is a really big one. But there's also IL-3, IL-6, IL-10, and all of these are going to be impacting or enhancing the impact of stem cell factor on mast cell growth. Mast cell growth is inhibited by certain things, such as GM-CSF, which we talked about in the last lecture, TGF-beta, various retinoids. And I really like this image over here because it sort of shows you how central this stem cell factor is as a cytokine for mast cells. It leads to not only survival and differentiation and maturation, but also it leads to degranulation and mediator release, which we'll get into. It results in migration and chemotaxis. So this is really a central mast cell factor that's really important to talk about. So let's talk about adhesion. Cellular adhesion molecule 1. This is pretty similar to ICAM1 and VCAM1, which we talked about previously. These are all just ways that cells, in this case a mast cell, which is the only leukocyte that has CADM1, is able to stick to things, and in sticking to things, it's able to infiltrate nearby tissues, so it's not just stuck circulating in the bloodstream forever. So this is an important one to cover because again, this is how mast cells infiltrate, and mast cell infiltration is one of the sort of key factors in a lot of allergic diseases. If we see that cells send out chemotactic signals that recruit mast cells, and those mast cells can recruit more mast cells, and that just leads to more and more degranulation. So the mast cells will have two intracellular sites that are required for binding. There's a firm binding motif, which anchors the CAD molecule to the cytoskeleton. There's also a PDZ binding motif, and this allows it to bind with the PDZ domains of other mast cells, or other things that are expressing that domain. Just talking about this a little bit more. There's a lot of different isoforms, not something we're really going to go into too much, but for example, the sp4 isoform, which you can see up here, allows mast cells to connect to other mast cells, and that's something we just talked about with that PDZ binding motif. It's going to allow them to connect to other mast cells and sort of form these mast cell aggregates that we see in allergic biopsies, for example. So this is relevant to asthma as well, because this adhesion molecule is what allows mast cells to sort of adhere to the airway smooth muscle, to infiltrate the smooth muscle, and we know that mast cells infiltrating the airway is something that's going to lead to airway responsiveness, for example. So now let's talk about how this adhesion molecule interacts with KIT. So we see that we know that this adhesion molecule and KIT are going to actually cooperate, and in doing so, they are going to promote the survival of human lung mast cells, or HLMCs. And so what does that actually mean? Well, when we upregulate or downregulate this adhesion molecule, it's going to have a mirror expression on the surface expression of the KIT receptor. So more of this adhesion molecule means more of the KIT receptor. More of the KIT receptor means there's more receptors to interact with stem cell factor, and that in turn means that we're going to have more survivability, more degranulation, more chemotaxis. So this adhesion molecule is not just about sticking to things, it's actually about being a co-stimulatory signal for other receptors that are involved in mast cell survival. So the theory about why this is the case is that this adhesion molecule helps to localize KIT to the surface of mast cells at the points of cell adhesion, right? So if this adhesion molecule needs to be on the surface to stick to things, well, it's going to actually help the KIT receptor sort of follow it to the surface, and that's what's going to allow it to interact with stem cell factor. So here's just a quick concept check. You can read this over and think for yourself, but based on what you know here, could you design some treatments for mast cell mediated asthma? Just hypothetically, what would you target? What direction would you want that targeting to go? And yeah, the answers are here if you want to pause. This is what I came up with, but I'm sure there's more than one way to go about it.